Why are biochemical tests so crucial in the field of microbiology? Well, these tests are the unsung heroes, providing invaluable insights into the world of microorganisms. They help scientists identify different bacteria, understand their characteristics, and even discover new species. Diving right in, the first test to discuss is the Gram stain. Named after the Danish bacteriologist Hans Christian Gram, this staining process is fundamental in bacteriology. It's a simple yet powerful method that differentiates bacteria into two broad groups, gram-positive and gram-negative. Here's how it works. The bacteria are first stained with a violet dye, then treated with a mordant, a substance that helps fix the dye. Next, a decolorizer washes away the dye, but only from the gram-negative bacteria. A counterstain is then applied, which the gram-negative bacteria pick up, turning them a different color. The result? Gram-positive bacteria appear purple, and gram-negative bacteria appear pink under a microscope. Now moving on to the catalase test. This test distinguishes between bacteria that produce the enzyme catalase and those that do not. So why is this important? Catalase plays a critical role in breaking down hydrogen peroxide, a potent compound that can wreak havoc on bacterial cells. The test is simple. When hydrogen peroxide is added to a bacterial culture, if bubbles form, it means the bacteria produce catalase and are therefore catalase positive. Next up is the IMVIC test, an acronym for indole, methyl red, Vosges-Proskauer, and citrate. This set of 4 different tests is used primarily to differentiate members of the Enterobacteriaceae family. The indole test checks for the ability of an organism to break down tryptophan. The methyl red and Vosges-Proskauer tests are both used to detect different fermentation pathways, and the citrate test examines an organism's ability to use citrate as its only carbon source. Following the IMVIC test, we have the MRVP test. The methyl red test checks whether the microbe performs mixed acids fermentation, and the Vosges-Proskauer test determines if the microbe is producing a specific fermentation product called 2,3-butanediol. Finally, we reach the protein carbohydrates tests. These are a group of tests used to determine whether a particular bacterium can utilize a specific carbohydrate or protein. This is done by inoculating the bacterium into a medium containing the carbohydrate or protein in question and a pH indicator. A change in the color of the medium indicates whether the bacterium can utilize the substance. In summary, biochemical tests are essential tools in microbiology. They enable us to distinguish between different bacteria, understand their metabolic capabilities, and identify their unique characteristics. From the simple gram stain to the more complex protein carbohydrates tests, each test provides a piece of the puzzle that makes up the intricate world of microorganisms. Biochemical tests, in essence, are the language through which we communicate with this unseen world.